Okay, good afternoon, everyone. Thank you all so much for joining us. It's my great pleasure to welcome back to campus Dr. Jaime Willbur. Dr. Willbur is a graduate of our chemical biology program. So she graduated from LTU in 2013. So just over a decade ago, she was sitting in those seats watching a seminar just like you, and now she's up presenting today. She majored in an interdisciplinary field, and she also uses both chemistry and biology, as she'll talk about, in her research and her career as a professor at MSU. So I had the great pleasure of working really closely with Jaime on her senior project. So she worked with me to express, purify, characterize an enzyme, and I could tell from the beginning she was meticulous, detail-oriented, and she was always curious. You know, in science, we always have to remember that it's an iterative process. Usually, you should all know by now maybe, experiments don't work the way you'd like them the first time, right? It's common to have challenges that you have to face and overcome, and Jaime was never deterred, and she was always curious and ready to try again. And you know, that's a great characteristic. I could see that she was going to be well prepared for graduate school. So Jaime worked really hard, fantastic senior project student, and she was actually a first author on one of my journal articles here, the first independent journal article I published at LTU. So, so I'm really excited that she was able to go on to graduate school. Between her junior and senior years, she completed an REU, an NSF program called Research Experiences for Undergraduates at the University of Minnesota during the summer. So she was paid to go do research there. And then she applied to graduate school, and she went to the University of Wisconsin, Madison, and focused in plant biology and plant pathology work. Right after she graduated in 2018, she was offered a job as an assistant professor at MSU, where she works in the Department of Plant, Soil, and Microbial Sciences. And today she's going to tell us a little about her career journey and curiosity science and how it relates to her career in agriculture. So thank you, Dr. Willbur, and welcome.
Thank you. All right, so I've got the microphone. I'm gonna not try to wander too far off the screen this way, but thank you so much for the introduction, and thank you everyone for having me today. It is wild to be back after 11 years. It doesn't feel like 11 years. I think being back on campus is interesting in that a lot of things are familiar, but there is so much that's been updated, and there are so many new differences and new improvements to the university. So it is fun to be back as well. Hopefully today we'll have a nice overview of how experimental curiosity has shaped my career in how I got from here, sitting where you are, all the way to where I am now at Michigan State University. And so we're going to actually go through, Dr. Timmons did a great job in outlining the different projects and paths that we will touch on today. And I will try to weave in throughout how those projects and those concepts still apply to everything that we do today in my research lab. And hopefully you'll learn something new. That's the goal today. So again, some of my background, starting off, I am a native Michigander. So I'm from Battle Creek, Michigan originally, Southwest Michigan, Cereal City, USA. I actually have a picture here of a satellite image of where I grew up. You will notice this is forest land, not farmland. So I did not grow up on a farm or associated with a farm. Farming and agriculture was not in my background, but I graduated from Penfield High School, if anybody's familiar with the area, and I had an interest in sciences in general, biology and chemistry. Living on the west side of the state, we benefited from the fruit agriculture, blueberries, strawberries, you pick, all that, famous things, delicious things. And my family promoted and encouraged just an appreciation for being outside, nature, national and state parks, and a love of travel and just doing new things. And so I think that helped set up a little bit of a foundation that helped to drive me through all of these different experiences. So, as Dr. Timmons mentioned, I was at the University of Minnesota. After I graduated, I was at LTU and graduated here in 2013 with a bachelor's in science in chemical biology. During my time here, I did an REU in Minnesota. Then I did my plant pathology PhD in Madison, Wisconsin, and then that led me to here. But here is our cohort of students, undergraduate students that I graduated with from here, and some similar faces. Dr. Zeeby, Dr. V, Dr. Timmons, and Dr. Morissette were our advisors at the time. I'm very excited to be back and still see them. And this is my graduate cohort. So these are the students and friends, still great friends that I graduated with from Madison, Wisconsin. We are now across the US from California to Delaware. And so we have a whole diversity of careers that we're in now. So even through graduate school, we ended up in very different places depending on our experiences and our interests. So here now at Michigan State University, I'm the research and extension professor in potato and sugar beet pathology. So this is really who is our program. All of these different faces, we have 4 graduate students, 2 senior researchers, a field and a laboratory technician, as well as a multitude of undergrads over the last several years, 2 currently working in our program. And each summer, we sometimes have the opportunity to have a high school student as well in our program. So we're working with a lot of different people. And in extension, this means, is anybody familiar with the term extension first? Dr. Zeeby? This may not be familiar to some. Extension is extending the knowledge back to our stakeholders. In my case, I have responsibilities with the potato and the sugar beet agricultural industries in Michigan. At many land-grant universities, the commodity groups were involved in forming the positions that I and my colleagues fill. And so they saw a need to have science at the university that would help them be better farmers and better growers. So my research program is developed from farmer questions and farmer interactions, and the results of my research goes back directly to those stakeholder groups. So it's an interesting back and forth. They're really interwoven in our program. So why Michigan agriculture? So some of you may not know, as I, when I was sitting in this room, did not know 11 years ago, the true diversity of Michigan agriculture. We're the second most diverse agricultural industry in the US. Thank you. And that's only after California, yes. So we have, spanning our state, our wonderful state, throughout the lower and upper peninsula, the industries of, we have livestock, we have fruit here on the west side of the state, we have my agricultural commodities are largely in the central area of the state, potatoes, and then sugar over here in the Bay and Thumb area. We also have fisheries, we have forestry, maple syrup. So it's a great, delicious place to work, actually. A couple of other things. Potato and sugar beet are both in the top 10 agricultural commodities in Michigan. And so my background is actually not in potato and sugar beet, even through my plant pathology, early plant pathology career. So these have been new industries to work with, and they're both active and wonderfully passionate industries of growers and ag representatives. So it's been a great learning experience. Another thing that we are actually first in the state is dry red cranberry dry beans for canning and drying. So there you go, that's something new, but we do have places in 1st, 2nd, 3rd, 4th, 5th, 6th, and 7th, and 8th throughout the US in Michigan's placement for an agricultural commodity. So what is plant pathology? So I'll put this here at the beginning, and then we'll travel there together. But Is there anyone who knew what plant pathology was before the seminar announcement came out, other than Dr. Zeeby? A little bit. So any guesses to shout out from somebody who was not familiar? Any guesses from what plant pathology is, or what a plant pathologist is? Any guesses? No? Yes? Yes, exactly. Identifying plant diseases. We are essentially plant doctors, right? Are a little bit different than human doctors, right? Our patients don't talk to us in quite the same way as human patients do. And we work with oftentimes many different crops and plant types. I work in potato and sugar beet, but also the commodities that those are grown with. And I have colleagues who work across the gamut of agricultural crops. So forests, trees, fruit crops, vegetable crops, greenhouse grown crops, all across that. And plant pathology involves, we have pathogen identification, etiology, or the progress of disease. We have different disease cycles. So Plant Path 101, disease cycles. So this is describing the interaction between the host and the pathogen, how and when the infection is happening, where the pathogen is living, and the conditions that arise to allow the disease to actually occur. We also cover economic impact, genetics, resistance, and host and pathogen resistance, as well as integrated management practices. This is the other second half of Plant 101, is called the disease triangle. So this is like the holy grail of plant pathology. We have three components that are required for disease to occur. That's a virulent pathogen, a pathogen that can cause disease, a susceptible host, one that can be susceptible to the infection happening, and a conducive environment, so the favorable conditions for both the pathogen to infect and the plant to be infected. And the cross-section of all three of those is when we have a risk of disease. So if you're not familiar with plant diseases, they may be closer than you think. So we have here just a few common examples of plant disease across the different categories of pathogens. So similar to human diseases, what are some examples of human pathogen groups broadly? Just shout them out. Virus, good job. What was that? Bacteria. Fungi, exactly. Any others? Those are the great three major ones. So we have on plants, same. We have a zucchini yellow breaking virus on pumpkins that often is contributing to the different colors or textures in different gourd groups. We have an example of a bacterial tomato bacterial spot. This is causing flecking on these fruits caused by a Xanthomonas species. We have an example of a fungal pathogen. This is Botrytis cinerea, which causes gray mold on strawberry, but also blueberry, grapes, lots of different fruits and vegetables. If you've ever driven by a corn, wheat, or soybean field and you've seen patchy yellow areas, this could be due to the presence of a nematode pathogen in the soil, microscopic worms. And the last one I have an example of is oomycete pathogens, which are fungal-like organisms. Phytophthora capsicea is the one I'm showing, but Phytophthora infestans actually was responsible for the Irish potato famine. And so if you have Irish descent, you are likely here in the US because of this plant disease, which is called late blight. So there you go. So what do plant pathologists do? So I work at a university, but we also work across many different sectors of agriculture and science. We work in agrochemical companies. I work annually with Corteva, Bayer, BASF, FMC, We have an agricultural research service, USDA unit, right in our department on campus. And we have many other regulatory agencies, nurseries and garden centers that we also work in. So just some examples. So a really brief overview now. So we're back to LTU. We're back to where we started. And I was advised by Dr. Shannon Timmons with the project to express, purify, and characterize sugar nucleotidyl transferases for use in the glycosylation of drug candidates. And so in this work, we the nucleotidyl transferases are used to synthesize sugar nucleotides. These are then taken by glycosyl transferases to attach monosaccharides from the sugar nucleotide donors to carbohydrate active drug molecules. This can increase or change the bioactivity of drug molecules, and this is an example of erythromycin A, which is a naturally glycosylated drug molecule used in lung or bacterial infections, chest infections for children, and such. So in this research, as an undergraduate, we worked to express, purify, and characterize these enzymes, specifically CPS2L, that catalyzes this reaction. And in this work, I really was exposed to an array of biochemistry techniques, typical laboratory techniques such as SDS-PAGE protein gel electrophoresis. We had HPLC-based assays to characterize the activity of these enzymes, and we quantified them using a protein standard curve-based Bradford assay. In my work now, I don't work so much with these types of enzymatic lab techniques, but we still do DNA, gel electrophoresis, we're still doing standard curve-based assays, so a lot of these concepts still apply. During this time, we were part of a group that was testing different temperature and times for incubating the expression of these enzymes, and we found that both did considerably impact the quantity of the enzyme that was expressed. So as Dr. Timmons mentioned, this was compiled into research. The first paper that I was actually on in biochemistry, molecular biology education. So the scientific writing experience, data collection, data analysis, the record keeping, all of those concepts were foundational to being able to survive and flourish in a research environment. As well as here, life at LTU was involved with both the chemistry and biology student groups. And this networking and outreach interactions that we were able to have, those were great for just keeping us active, learning from each other, studying with each other, surviving together, and all of those team and networking activities still took me through graduate school and into my current career. So back to experimental curiosity. So I have designated the time at LTU as really foundational in developing the skills of how to ask and answer questions. So this is based in principles, basic principles and applied principles in science, technology, math, and leadership. The laboratory practices in chemistry and biology, data analysis and scientific writing. Again, there are some activities that we did is in report writing that I still refer back to for students that are in my research program now. Those are critical skills. Individual and group learning experiences, and the start of professional development, networking, and outreach activities. And again, we talked about the REU experience. So in the summer of my junior year, I had the opportunity to go to the University of Minnesota Twin Cities. I was in a lab located on the St. Paul campus, and this was with the Life Sciences Summer Undergraduate Research Program, LSERP, because that's a great abbreviation. And in this, so I would say in this program, I was matched with a plant pathologist because of some of the lab experiences that I had had. I did not at that time yet know that I would enjoy plant pathology as much as I did. So being curious and open-minded enough to try it and to be open to the process of this new lab and this new research program was obviously critical in my own personal and professional journey that had led to where I am today. So in this lab, my advisor was Dr. Deborah Samick. She is a research plant pathologist specializing in alfalfa fungal pathogens. And our project was the effects of foliar fungicide applications on alfalfa pathogens. So this is an alfalfa field here. From the road, it might look pretty green. To a plant pathologist, we might start to notice some of these little yellow areas here. Yellow to a plant is less chlorophyll, less green, less photochemical activity that's going on. And up close, we can see that's because of some of this necrotic area on the plant caused by some of these fungal pathogens. So Dr. Samick's program focused on disease resistance, genetic defense mechanisms, improving nutritional quality of alfalfa crops, and management strategies. So why alfalfa? This is the primary forage crop for dairy cows. This is a high protein and fiber content crop, and the leaves are the major source of protein. So that's important for us. So trying to relate this back to you, by show of hands, who enjoys dairy? Ice cream, cheese, milkshakes, all that good stuff. So these numbers are a little outdated, but based on 2012 numbers, per person in the US, we consume about 612 pounds of dairy per year. This, supported by the population numbers at that time, and the amount of milk that it requires to produce that much dairy, if we do some back of the envelope calculations all the way down to how much alfalfa is required per cow, then how many cows we have in the US, we get to about 50.5 billion pounds of alfalfa hay required per year to support our personal dairy preferences. And this checks out. There's about 60 billion pounds of alfalfa that is produced each year, all in support of our consumption of cheese and milkshakes, my consumption of cheese and milkshakes. So foliar diseases then. So, right, the leaves are the source of protein for these cows, and we see that in healthy alfalfa, lots of leaves on this stem. In diseased alfalfa plants, though, these foliar fungal pathogens start to kill off these leaves and the plant drops them. They're of no use any longer. So the defoliation can be up to 70% in some cases, and this reduces overall crop yield, but also forage quality. In this specific case, we were looking at two foliar fungal alfalfa pathogens, Stemvelium leaf spot and black stem and leaf spot caused by Stemvelium botuliosum and Phoma medicogenus. And the chemical component of this project was focused on one fungicide. This is a headline. This is a BASF product. This is still applied today. I still have growers in sugar beets that use this product. And this active ingredient, especially in this group of compounds in general, are still widely used in agricultural disease management. This active ingredient, pyraclostrobin, is a carbamate ester that is targeting the electron transport chain in the fungal mitochondrial membrane. So right here, this is a quinone outside inhibitor. So in this complex 3, this is blocking the electron transport, which ultimately stops the energy flow or energy production for this fungus. killing these cells or spores that this product is applied to. While strobilurins, I would mention, aren't commonly used in human medicine, triazoles, which are another group, are commonly used and widely throughout agriculture as well as human medicine. So our objectives here were to evaluate alfalfa fungicide treatments for control of fungal disease and reducing the amount of defoliation. So, right, our hypothesis here is that when you apply a chemical treatment, you're reducing the amount of disease and you're promoting yield. The characterizing fungal pathogen sensitivity is in the lab. So this is a lab-based assay. We were testing pathogen responses in a germination assay. So we amended artificial media with this product, and we were assessing the percentage of those spores that could germinate in the presence of that chemical. But yes, this project had visual evaluations. These were samples coming from the field. We processed them in the lab, isolated fungi from them, sequenced the DNA to confirm identity, and then did these in vitro sensitivity assays. The results from this experiment were that across 5 different locations in the Midwest, we did see a significant reduction in this teal bar, are the ones that were treated with this headline fungicide. So that's a reduction in percent defoliation. So we did see successful control. After my time there, they also assessed the yield. So this was associated generally with yield increases. So reduced disease and increased yield. That's both important. And then also, this was a large portion of my project there during the summer, was for each of these different fungal isolates, we were doing this plate-based assay. And these results are reporting the concentration at which 50% of fungal growth is inhibited, or conidial germination, spore germination in this case. So what we found, we were expecting that resistant isolates would have between a 2 to 44 parts per million response. We're at about 0.05 to 0.06 in this case. So in this case, we did not identify resistant isolates, and we didn't identify a difference between these 2 treatments, which is a good thing. In my current position, we still are monitoring for fungicide resistance to about 8 different active ingredient compounds for a couple of different fungal pathogens. Some of that we do see up to 100 parts per million responses, resistance responses. This information, though, helps growers to decide what chemicals they might use in the field. So we deliver that information back to them annually. Some of what we do now does incorporate some known genetic mutations with that fungicide resistance or pesticide resistance. And so some of it is a little bit more high throughput. Some of it is exactly the same as this was when I first learned it. Back to experimental curiosity then, this experience was really expanding my horizons. So I was learning the principles of plant pathology and microbiology outside of general bio and general chem, but I also exercised the scientific method more. I became fascinated with this interface of science and agriculture, and also an appreciation for applied research. So being able to receive these samples right from a farm, right from a research farm, handle them, take data from them, and that data goes right back to those farmers. And connecting with diverse audiences. So remember, this REU program was not just plant pathology. This was across all life sciences. So we were presenting research to each other and trying to translate what we were doing and how it was important to any other area of expertise in science. And this was, of course, continued professional development. If you're interested in research and continuing on in academia, write write. Just keep practicing writing, because that is a developed skill and it takes time, and all of these experiences just added to that. The data collection, the analysis, the translating that into writing, and oral presentations as well. Those are all transferable skills. This also resulted in actually 2 publications during my time after my time at University of Minnesota, which was also excellent experience to learn a little bit more about the research publication realm and how to navigate that in reviews and reviewer feedback. So all of this led to me jumping onto the plant pathology train and deciding after I graduated from LTU to pursue an opportunity at the University of Wisconsin-Madison in plant pathology. So this was a direct admit PhD program, which means that I did not have to stop for a master's degree. I was committing to a full PhD program, as long as they would have me. And so this work focused on soybeans and a fungal disease called white mold. In this picture, this dark green back here, relatively healthy plants. This lighter green, yellowish color up here are all plants that are impacted by white mold. And this project was a collaborative project across many different states, soybean growing states and universities. So soybeans then are grown for meal and oil. 80% of the soybean is meal, 20% is oil. Most of the meal goes to animal feed, and most of the oil actually goes to food products and biodiesel. In the US, we are the top soybean producer in the world, followed by Brazil and Argentina. So this has wide-reaching impacts wherever this crop is grown. Oftentimes, these diseases are also an issue. White mold is one of the top soybean diseases, especially here in the north central US. So starting with, let's do a little diagnostic exercise then. So in this picture, what do you see? Do you see anything that might be concerning if you were a soybean grower or a soybean farmer? Yes. Patches of the lighter green leaves, right? We have maybe here, if you look closely, some of those leaves are curled and starting to kind of wilt a little bit. A little bit here in the front center, we have some that are more necrotic or brown. So we're changing from yellow to brown, which is more severe. Those plants are dead and not likely to come back to life. So when we take a closer look, and we plant pathologists have to do this every day, we get sent these samples or photos of samples. We have to ask more questions on what is that symptom from? Is it from not enough water? Is it from the wrong nutrient combination? Or in some cases, it is from a disease. So down lower on the stems, if you open up the canopy and take a look, these stems are colonized with this white mold. This is a fungal pathogen that causes, once it infects, it actually hollows out the stem and clogs it with fungal tissue. So it's like clogging a straw. So no water, no nutrient transport, and that's why we get the wilting at the top of the plant. Now, as a grower, you see the wilting on the top of the plant. You're driving by 50 miles an hour on your country road, and you see this happening. Unfortunately, that means that the fungus is already infected, already colonized the stem, and already resulting in yield loss. You'll notice these are soybean pods here. This is all at the yield, priority yield components of this plant. This can prevent pods from forming. It can actually colonize pods as well. And so all of this is inhibiting the ultimate profit of these farmers. So this pathogen, again, life cycle, we have the fungal pathogen is actually overwintering as a survival structure in the soil. This is a dormant structure. It can wait 5 or more years in the soil until the right conditions arise for it to germinate and form these cute little cup-shaped mushrooms. These are called apothecia. They look like a little shallow martini glass right on the soil surface. In cool, humid conditions, these release ascospores. They infect the flowers of the soybean. These serve as a nutrient source, jettisoning this fungus into the stem area, colonizing the stem. And at the end of the season, once the nutrients are spent, these dormant survival structures are formed again throughout the stems. And unfortunately, when the grower comes through and harvests, all of those survival structures are fireworked out back into the soil to be incorporated and survive another 5-year cycle. So in plant pathology, we use disease cycles as inspiration for how to approach the management question. So where in this disease cycle are there opportunities to stop the cycle, essentially? So in here, protecting these flowers from being infected, that would be great, that would be key. Reducing the amount of these sclerotia that are formed and overwintering. So in this project, and as a graduate student, we focused on some of these farmer questions that we were being asked. So commonly, can I apply fungicides when I first see the disease at that canopy level? Unfortunately not, for the reasons we explained. But if not, when should I apply? Fair question. It's when these little mushrooms are out releasing the inoculum to those flowers. Now, this is a dime for size comparison, and these are the apothecia. Right here on the surface of the soil, these are smaller than the pencil eraser usually, and depending on the soil texture, can be very similar in color. So even as graduate students doing a lot of grunt work going through the field, sometimes we had a really hard time finding this smaller than a dime structure in a 50 to 100 acre field. It's a lot of ground to cover, and the growers also are dealing with, is this the white mold mushroom? Other fungi exist in this environment, and they can look similar. This is also described as a cup-shaped fungus. So being able to distinguish between this as a pathogen and this as a saprophyte, which is just living on dead plant material passively, not causing a problem. So this research question then is, how can we support producer decision making? What can we do to help them in this process to make biologically informed management decisions? So our objectives were to improve risk prediction by modeling the presence of apothecia. Is there some way where we could have an app or a tool that alerts them when there's this potential risk of these mushrooms being present in their fields? And then to use this information to help them apply these chemical treatments. Based on that risk, what is the recommendation? And just a note, most fungicides should be applied before the infection occurs, so we really are timing before we start to see disease on the plants. So in this project, and this is just an example of this type of work in plant pathology, the types of methods and techniques that we use for predictive modeling work. This is an epidemiological study, so we're looking at how this disease is forming and progressing, and we are often intensively scouting and monitoring in multiple fields across multiple states is always better, multiple years. And we had trials in Iowa, Michigan, and Wisconsin. I was mostly managing the Wisconsin trials and all of the data generated from the other states. We collected these observations of this fungus in the field, as well as gridded and on-site weather data. So now we're tying in meteorology, so climatology, engineering, and technology in these Campbell weather stations. I was involved in wiring these to data loggers. These are sensors that are collecting data remotely for us, and this was an annual thing. We had to reset these up, but these are the exact type of weather stations that the weather programs would use, meteorological programs would use. So in this model development work, we are considering the relationships between the pathogen, the actual biology of the pathogen, and weather conditions. So in this case it was weather conditions. We're looking at air temperature, relative humidity, rainfall, but this could also be, and this is the work we do now, is it wind speed? Is it particle dynamics, flow dynamics? Is this the physics of a spore and how it moves through the air and how it's deposited out of the air? These are all parts of how this pathogen is being successful. And so this could, yes, these relationships are then, we look at them through regression analyses. So anybody remember the equation for a simple linear line? The equation of a line. Y equals? Yes, mx plus b. So in this, we have y, which is our mushroom presence or abundance, the numbers of those mushrooms. In mx, so our x variables then, we have multiple options and possibilities. And so they might be the air, they might be the physics, they might be the collective hours that you all slept the last 30 days. It could be anything. And we are going through statistical modeling processes to select the best relationship of the most explanatory variables. Within reason, we are somewhat limited by the amount of data that we have or the types of variables that we can collect. So in plant pathology modeling, we're aiming for an improvement, something that is better than just a coin flip of yes, it's going to happen, or no, it's not. Is there something that we can help the grower just a little bit more understand this process? So as a graduate student, we were able to develop 2 different models, one for irrigated production systems and one for non. And in this case, we actually did have scenarios where our model, which is this line, was below our risk threshold. This is if a grower was to apply a treatment, it would have to pass this red line during this green box, the susceptible period for soybean. So we had scenarios where it was below that line. So the grower, in this case, a soybean grower, could have saved a chemical treatment. Let's say a grower has 500 acres of soybeans. Even one application is about $45 an acre. So we're talking $22,000 of savings by just not applying once. And so these are valuable decisions that these growers are trying to make. And if they did have to apply, we found that this model helped them to time the application within this risk period, earlier or later, to make the most effective treatment application. So across multiple states and multiple sites and multiple years, we had about an 81.8% accuracy. So in this case, better than 50 in the 80 to 90 range is great. Again, this is not a silver bullet tool. This is an integrated tool, part of the toolbox that a grower might use. These models were actually developed into iPhone and Android app, and I have to thank my advisor, Dr. Damon Smith, who's continuing this work at the University of Wisconsin-Madison, and we have more involvement in the irrigated system portion of this model as well, still here in Michigan. This app, this is the Android version of here, is using our validated models. It's available in the US and Canada, and it uses grower input. So a grower would add in some information about their fields, and then it pulls weather data, does the calculation, and puts out the risk threshold, the risk percentage for that field area. So here, we'll walk through it. So this is where a grower would add some of the details of their field. They would talk about soybean flowers, canopy closure, and then it would pop out this risk calculation. So if the risk is medium or red, they can choose to make an application based on that decision, based on this information. Or if it's low, they might choose not to. But they can save this information, they can save multiple fields, they can rerun this at multiple times throughout the year. And this app is actually being used. So this is getting thousands of downloads now. And soybean growers are referring to this to help make those 1 or 2 applications for this particular disease. So that had an extension award, which was great for the University of Wisconsin-Madison team that's still managing that. But in this project, we kind of learned the qualities of connecting and collaborating, right? We're connecting with our growers. What do they need? We're collaborating with multiple researchers. This is not an individual effort, and we get so much more done together. So that's something that we continue to try to strive for today. And yes, all of the funding and acknowledgements and colleagues that supported this project. So that brings us to today. My position currently at the Michigan State University Potato and Sugar Beet Pathology Program. Again, you'll have noticed there was no potatoes and no sugar beets in the last several projects that we worked on. And that's okay, because some of these skills were transferable and a lot of these concepts are transferable, and we're still working on predictive modeling. We're still working on fungicide sensitivity, chemical management in the field. And we have many different collaborators and funding groups, including the USDA, our stakeholder commodity groups, multiple other states of research colleagues, and this would not be possible without all of them. So, bringing it back to potatoes. So potatoes in Michigan. Potatoes generally are the most important vegetable crop globally. Incredible source of carbohydrates and nutrients, and it is, Michigan is the 8th largest potato producer in the US. Like 8, but we are number 1 in the nation for potatoes that are produced for chip processing, potato chips. So who likes potato chips? Yes. So potato chips, right, think of that typical Lay's bag of potato chips, the original kind. They're round, they're about this big, and they're pale yellow. That is the ideal potato chip. In Michigan, our potatoes that we grow are round, white-fleshed potatoes, ideal for slicing into potato chips. I am in the perfect position in potato research because potatoes are my vice, so I just feed my vice. In Michigan, we have approximately 50,000 acres of potatoes planted, and this is generating around $2.5 billion in local economy and jobs and throughout the potato production system. So in this map, we can see potatoes are produced in over 40 counties of Michigan through the lower and upper peninsula. I'm in the upper peninsula approximately 2 times a year, 3 times a year. It's a beautiful drive, and most of our research is actually centered in the Montcalm County region. This is a central sandy area. Potatoes grow very nicely there. And another fun fact is that we have about 1 in 4 bags of chips in the US are filled with potatoes that come from Michigan. So that's an incredible contribution to chip production. And in the state, we have Better Made and Great Lakes chipping companies, which you may have enjoyed or heard of. If you have noticed, this is another connection, any of the purple potato chips that are produced by the Great Lakes Kettle Cooked Chip Company, these purple potato chips were actually, the potatoes were developed at MSU, at Michigan State University, in the potato breeding program. They're called the variety Blackberry. These potatoes are purple, dark, purple-fleshed outside and inside, skin and flesh. And this is approximately the color on that bag. So the interesting thing with these is that even after frying, they stay that nice dark purple color. So if you enjoy those, think of MSU, shout out. And my colleagues there in the potato breeding program. This, in Michigan, we do have a potato industry commission, which coordinates both funding and research opportunities between the university researchers and the growers. They also are coordinating marketing and education opportunities with the public and with government. And so this is our Potato Industry Commission. One of their initiatives is to help you as consumers connect with the producers, the potato producers. So if you ever see in the store Michigan potatoes, blue Michigan potatoes tag, you have the opportunity to enter in those number characters, and it will take you to the potato farmer that has produced those potatoes. I probably work with those farmers, and they greatly appreciate knowing that you all can see that. And so thank you if you do. On the other hand, sugar beets, right? So sugar beets, has anybody ever seen a sugar beet? Yes, some. Okay, as an undergraduate student sitting here, originally from Southwest Michigan, I did not know that we grew sugar beets in Michigan. I did not know that. So don't feel bad if you haven't. We have 150,000 acres of sugar beets planted in Michigan. We actually produce 1.2 billion pounds of sugar per year through Michigan Sugar Company. And I didn't know that even existed, even through most of my graduate school. So my work in in soybeans and alfalfa, that transferred to sugar beets, even though I didn't directly work in them. But coming back to this state and being able to appreciate some of these other areas of agriculture that as a native living here, I didn't previously, has been a really nice eye-opening experience. And Michigan Sugar Company, so this is our sugar company, is actually located in Bay City. This was founded in 1906 and is the third largest sugar producing company in the US. Other regions include Idaho and the Red River Valley in North Dakota and Minnesota. But we have approximately 20 counties of sugar beet production. And yes, we're slicing about 22,000 tons of beets per day to meet this production goal. This sugar is packaged as Pioneer or Big Chief sugar. This is white sugar, brown sugar, and the powdered sugar, the other one. And this all happens within this area of the state. So actually, our sugar beet production or our sugar beet research takes place near Frankenmuth, Michigan. So we get to drive through Frankenmuth every time we're out there in the summer. And this is at the Saginaw Valley Research and Extension Center. So they're off-site research centers that Michigan State University funds and runs. But we are there probably 1 or 2 times a week during the summer. So it's a 3-hour round trip to most of our off-site research areas and trials. Oh yes, in the US, just to mention that we have over 50%, 55% of our sugar on the shelves is coming from sugar beets. So globally, it's about 1/3, but here in the US, it's over half. And if you look at your probably generic brands, Meijer, Walmart, if you look on the bags, it probably says sugar from sugar beets. So across these 2 crops, we are working with a diversity of diseases across viruses, bacteria, fungi, oomycetes, nematodes, the whole range. And we are lucky in that our crop, the most important, valuable part of these plants, is grown in the dirt, underneath the ground, with all of the other microbes and wonderfulness that is in our soil. So we have diseases that are originating in the soil, in the seed, and also impacting the above ground parts of the plant, the leaves, the stems. And in Michigan, we have niche agricultural industries for potato and sugar beet that continue past the typical growing season. So when we harvest potatoes and beets, actually quite a lot of that crop is stored throughout the winter so that you can continually process for when the companies and processors need them. So potatoes we can store up to 7 months. So up to the next June, we have potatoes from the previous season. In sugar beets, it's usually till about April or so that we still have sugar beets from the previous year being sliced and sugar extracted. So we actually have post-harvest disease research as well. So starting from planting all the way through the storage period. So in my program, actually, we work a lot in field in the summer, but our experiments are continuing all year round. They just change the nature of them slightly. Everybody in my program has their own independent research project, but we're working together for major activity days like planting, harvesting, data collection of the crop, winter experiments, all that jazz. So it's a nice dynamic environment for all of us in the program. One of the examples of research that I'll leave you with from my MSU program is led by a PhD candidate, Alexandra Hernandez. She's aiming to finish up this summer. She'll be the first PhD student in the program to graduate, and we just had a master's student finish up this semester. So it's been a wonderful experience, and I've been really fortunate with just the personnel and the students that have come through the program and their resilience through COVID and a new program through all of that. So her research is supported by different sugar beet funding agencies and MSU funding agencies, and it's focused on Cercospora leaf spot. So this is another foliar fungal pathogen. And this overwinters actually on infected leaf material. So these cause this leaf spot on the leaves. And these little black dots, those are similar dormant survival structures to the first pathogen that we talked about. They contain melanin, so that dark color actually makes them more resilient to temperature fluctuation and rain, moisture fluctuation. In the following, I should explain sugar beet harvest. So sugar beets have nice foliar leaves that are above ground. The roots are what we are harvesting. So right before harvest, the leaves are actually cut off and removed from the plant. Those are usually left in the field. So any infected leaves are returning to that field soil. If you incorporate them into the soil, they have a larger chance of breaking down. But with our new sustainable initiatives over the last decade, we have moved towards minimal tillage, so less soil disturbance, but also that means that we have more pathogens that are overwintering. And so that is a problem for our sugar beet growers. Cercospora leaf spot is the major foliar pathogen of sugar beets wherever they're grown throughout Europe and the US. And this pathogen, one of the reasons it's such a problem is that they germinate in the spring and they are sporulating. So they're releasing these spores. These spores can infect new leaves and then reproduce within about a 10 to 12 day span. So that's every 10 to 12 days, starting about June, there's a new generation of this fungus that's re-entering the production system. So it's hitting an exponential growth phase actually until you run out of plant material essentially. This in terms of chemical treatment means that we're trying to protect the leaves every 2 weeks once it gets started and established. So we are dealing with fungicide resistance across several different product groups. That's something that we annually report. But for Alex's project, we're looking again, where are other opportunities for management? So if not here, if not protecting leaves, we wondered about this overwintering survival. So if tillage is maybe not an option because we don't want to promote soil disturbance and erosion, what else could we maybe do at this point to reduce the amount of survival and reduce the amount of inoculum potential the next year? So in this project, some of the background information is that aside from overwintering on leaf material, this particular pathogen is sensitive to temperatures over 100 degrees Fahrenheit, which Anybody here is like me, we understand this completely because we are also fairly sensitive to over 100 degree temperatures. And so we aimed to utilize that information and look at different ways that we could target the pathogen at the end of the season into the following spring. This is essentially a long-term reduction of inoculum. So here, I'm showing a propane-fueled burner. So this is very flashy, fire, woo. But there are flames here, but actually we're not burning the leaves. We're not torching them to a crisp. We're heating them at a high temperature for a very brief amount of time as we're driving through the field. If there's infections on those leaves and they're exposed directly, then we're defoliating. Those treated leaves are going to sit on the surface of the soil, and then we wanted to measure how much pathogen is coming from them the next spring. So these are inherently long-term 2-year projects just to get some of this data. And we had, featuring some of the technicians and researchers who were driving the tractor, helping to fabricate this propane burner and run it. What we did find was that we did see in these heat-treated plots, that over the winter, we were able to see significant leaf degradation, which is similar to if we had tilled or plowed. So that's great. We see leaf degradation, so degradation of the environment that the pathogen is in. And this was significantly different from our control, non-heat treated. This was also monitored in the actual spore levels coming out of that soil or out of those leaves. So we saw significant reductions in the amount of spores for up to 2 months in the beginning of the season. So again, we saw this suppression in these 1 and 3 mile per hour heat treated plots and continuing through the full season. We didn't apply any other management to those plots. We were able to identify suppression of disease in the absence of any other type of management in those heat-treated plots, similar to our tillage treatment again. So this is interesting, right? This is a different type of approach for our growers. And while we may not be recommending every grower out there to be flame torching their crops, it's a new concept to revisit about what does this type of management look like at the end of the fall through the winter to the spring. And again, it's not a silver bullet, but this reduction could be combined with other management tools to then layer into more and more reduction of disease without the added pressure of fungicide resistance. I would mention here, this is area under the disease progress curve. Calculus, anyone? This is an applied version of a simplified calculus where we are measuring disease over the season and we're summarizing it in a single value of disease pressure based on the AUDPC. So shout out to my calculus professors here for that. And so experimental curiosity again. Encouraging everyone to just think outside the box. This sounds like an off-the-wall type of approach, but in high-intensity systems, sometimes that's where we're at. And to stay informed, but still think creatively, because you have to know where the box is to think outside of it. And then that brings us to the end. So I'm just summarizing here the general ideas of experimental curiosity. In my own personal and professional career, this brought me through research experiences as an undergrad, all the way through postgraduate into my current faculty position. This continues to drive our program activities and research and extension every day. Every day we're using this, we need to stay curious and stay inspired. And so this is fueled by many different sources of inspiration. This can come from our growers, the students in my program, peers and colleagues, but also family, home, mentors, your personal hobbies or personal beliefs and values. Like all of that goes into staying fresh, being able to think creatively, and stay curious. Our research program involves diverse approaches to answering these questions and collaborations as well. And this spans, hopefully you've been able to get a glimpse of this spanning agriculture, crop science, chemistry, biology, microbiology, math, statistics, technology, economics, as well as social science. So everything that we do, it still comes down to the grower choice and adoption and use of those practices. So that's a social science element that sometimes we are working with other researchers in that to better understand decision-making patterns. And every day we are learning something new. I am learning something new. I have learned many new things, and I continue to do so. And I think that helps to keep us active. It helps to keep us flexible, and it helps to keep us relevant by interacting with our growers and keeping open-minded to their questions and being able to learn with them. So that would be my last piece of advice. Of course, I thank all of the members of my program, all of our industry stakeholders and collaborators, as well as funding sources. With that, I'll mention that some of my inspiration is our home hobby farm. My husband and I got married in 2021, and we do have a hobby farm with chickens, ducks, geese, 2 pigs, many dogs, and cats. And so this is just part of being able to do what I do every day, is having this at home, keeping me going. So thank you. Again, it's been a pleasure to be here.
Thank you, Dr. Willbur, for a fascinating interdisciplinary talk. At this time, I want to make sure everyone has a chance to eat pizza, so I welcome you all to enjoy some pizza in the lobby. Dr. Willbur is going to stick around, so please, students, introduce yourself, ask your questions outside, and enjoy the rest of your day, everyone. Thank you for coming.